So it's really nice to be here with you today. We prepared material in 2 blocks. Today we would like to discuss the interactive spectral indices, and tomorrow, same time, we would like to discuss optical quality estimation, introducing perClass. So perClass is a Dutch company. We are based in the city of Delft. Originally, we did research in machine learning and spectral imaging, and we focused on industrial applications a number of years ago. And then in 2018, we brought specific product for spectral imaging market. That is the perClass Mira user interface. And what we try to do with this interface is to enable people in the industry in applications to basically analyze, interpret spectral images without machine learning expertise and without programming, very interactively, and deploy these solutions into different types of applications. So sorting applications, phenotyping applications, and so on. And we will be using this software in the tutorial. So for us, the major question we are addressing here is, if you have spectral cubes and in your own specific domain, it can be, let's say, grading application, food quality application, plant phenotyping application. Then there are many questions that you need to address if you want to go from the cube to the interpretation. And that's starting with what sensor technology you really want to use, what wavelength range is important for your application, how do you correct the data, how do you annotate, how do you learn from this data, different statistical models, and how to set up experiments, and how to actually get something meaningful. That's what we are trying to address here. What we mean by spectral image interpretation, of course, there are many different ways to look at it. We roughly speak about 3 different things. One is classification. So we will see how to learn how to apply classifiers, how to make decisions on the pixel level, on the object level, how to deal with applications such as foreign object detection. The second one is, let's say, visualization and feature extraction. So how do you get meaningful information from your spectral data, typically only on the objects of interest, let's say disease patterns on the plant or stress level of the plant and so on. And the third one is regression or estimation, where we try to estimate some quality characteristics such as sugar content from a tomato or the dry matter content in a tomato, and so on. So these are roughly the areas, and today we would like to focus on classification, visualization, and tomorrow on regression. So the software actually is working, or we are working with many different types of cameras, and you can also use just generic NV cubes or MATLAB cubes or TIFF stacks. So it's not really limited to a specific sensor, but with our customers, we actually see often these types of cameras used from really different domains, line scans, snapshots, systems with few bands, systems with hundreds of bands. So that's actually quite varied. Here you can see the initial screen of perClass Mira. We can select different project for specific camera types. We have presets, but here we can use just NV cubes, and we also can select the top-level data directory where the data is actually located. So in the downloads that we provided for the first tutorial, you see 2 data sets, potato virus and tulips. So we just select the potato virus directory. If you have on your computer a GPU, NVIDIA GPU supporting CUDA, you can select it. In the installation of perClass Mira is actually a separate executable that can deal with CPU or GPU. So you can use the GPU. Okay, we have this environment, and the idea is that we can add one or more images to our project. So let's go to the images list on the left side, and we right-click, context menu, add images to project. And here we see the HDR files, so the header files for the NV cubes that are in our data directory. So we can select one or multiple of them and load the cubes. So what you can see now is that we have 4 scans of potato plants. This is data coming from our customer, Wageningen Research, doing different phenotyping studies. These are hyperspectral cubes. So if we move with our cursor over the image, on the right side, you can see a spectral plot where we can see that the data is from 400 nanometers to 1000 nanometers. So it's in a linear range. And we can go through this cube just by dragging this blue line. The reddish part, it's just a visualization thing that is basically showing us that pixels in this area are above the upper bound of the plot. So it's not changing anyhow the data. It's just adjusting the range. And this is a single band view. We can also change to the preview where we get 3 lines for the RGB, and we can just drag them. So for Wisner data, you can very nicely get more realistic color images of your sample, like for example here for the plants. Now in this problem, we have 2 healthy controls, and we have 2 plants that have virus infection. What we would like to do in this tutorial is first of all learn how to build a classification solution on the pixel level, and then we will look at the visualization and interactive construction of spectral indices, and then we actually use both of these topics or technologies together. So in projects, let's say phenotyping projects, the first task will be segmentation of a plant from background. And what we can do in perClass Mira is just define some classes. You see the list of classes on the left side. We can say a new class, and we can just type say, background. We could select a color for that class, and basically you can just paint some examples of the background. And we create another class called plant or leaves, let's say. And we can select or we can paint some examples of the leaves. Now, on the right side, in the spectral plot, you see that when we paint, we get the spectral plot updated. We can see the mean of the class, the pixels, and we can see the min and max. We get the feeling for the range of values. If we want to zoom inside the image, we can use zoom in and out button, or simply hold Control key and then zoom to the point. So it's very easy to get closer. We are not interested only in the leaves. In many phenotyping projects, scientists need to understand the difference between the leaves and the stem. So we define another class for stem, give it a different color, and provide some examples of the stem. Okay. Then we basically want to build a classification model. We can do it by clicking the model search on the toolbar. And what happens is that the software is using the labeled information to build statistical model and then apply it to the entire image. So we are still looking at the same image. We were on the layer of labels. So that was the information we provided. And then when we train a model, we get to the layer of decisions, where for each pixel, we get one of the classes as decision of the classifier. We are blending the 2 layers, so there is an alpha button on the toolbar, and in one extreme, you can see that we only really have the decisions. So that is really the output of the classifier under the decision. and we are blending it with the underlying image. That helps us sometimes to understand better how we do actually in practice. So the idea behind classification in perClass Mira is that you can very easily define some problem and define labels and then build a classifier and improve it. So here we can see that, for example, at the end of the image, we have some, let's say, this should be background. We can try to just train the model, and often that is one quick way that, that helps us to improve. Another thing that we can do is we can engage a tool that is actually doing something called active learning, so that we can enable this show unknown tool on the toolbar, and you can see that there is an extra like a gray or a transparent set of pixels. These pixels are very different from what we labeled before. So if we want to label somewhere, we should label in these places. So we go back and we, for example, say this is the leaf and retain the model, and in that way we improve. Or here we see that we have a stem that is going up so far, and basically we can provide the relevant labeling. So this is should be actually a leaf. I labeled with a different class. You can simply select the class, right click and say assign the region to a different class. So there are a number of tools that allow you to improve the labeling and improve the modeling very quickly. So again, right click. So if you want to switch between the classes, you can just use the digits. There is a lot of different keyboard shortcuts because you want to really focus on the problem and not on the tool itself. We may want to improve also the background segmentation and maybe add a label class that we also want to segment out from the images and build a different model here. Now, well-trained model, classification model, we get a well-trained model when actually on relevant data, you will see very little amount of this not known decisions. And what we should do is to really look at different scans, of course, and then use them for learning. So we could also, of course, start learning from another image just by labeling in the other image. All the data that you label in any of the scans is used for building the model. We may also want to use some of the images only for testing. So you can right click and you can just flag some of the images for testing. So these green images will never be used for any kind of modeling, and it gives us, if we actually retrain again, we don't use those samples in the training process, but we can get estimates, or we can see how the models are really working on unseen data. So there is much more to classification than this. I'll just show you quickly that you can really drill down. You can, for example, see the confusion matrix. So these are actually for each class the pixels that we labeled in the rows, and in the columns the decisions that the model is doing on the training set and also on the test set. So we only labeled some examples on this second image, and that's why we have here only 2 classes present. But this allows us to basically understand where the errors are happening. Now, we don't have only healthy plants. We also have the sick plants, let's say. And you can see that in this scan, we are seeing some extra patterns. There is actually a virus infection happening, and that's influencing how the plant leaves are affected, and we might want to define a specific class for this pattern as well. And gradually we get to better classifier. We may also want to remove the background here if it's possible. Another thing that I would like to show you is that we don't need to use the entire spectrum. In this data, we have 192 spectral bands for each cube. The cubes have 192 spectral bands. But we may freely choose what information to use. We can, for example, say that we don't want the ends of the spectrum to be used for the classification, retrain, and basically only use the green, the enabled bands. You may also remove everything and really define very specific ranges. And in this way, you can find out when things break and what information you actually really need for the classification. So this is helping when you want to decide which sensor you want to use, which range of spectra is more informative. So what we basically did so far is we built a classifier on the pixel level, and that's a good start. Now I would like to shift to the part of the visualization and spectral indices. So let's actually go to the visualization panel, and I just reorganize these panels that we have a visualization on top of the spectral plot. Here we can choose different common equations that are used, spectral indices, and if we select any of them, for example, here it's just a division between the two bands, you can see the definition, the upper panel, and then in the spectral plot, we actually see the spectral range that is forming the first and the second parameter, so the numerator and the denominator. You may remove all the extra information in the plot and zoom a little bit. So what we can actually do here is that we can see interactively what happens if we compute, if we integrate the reflectance in each of these spectral areas, and then we divide the two numbers. Now that's interesting, but of course it's not so informative because we see a lot of response from even the pot of the plant that is not interesting for us or from the plastic label. But we already built a classifier, and because we have a classifier in perClass Mira, we can also work not on the pixel level but on the object level. So if I go back to the decisions, I can actually flag some of the classes that are of interest as foreground. And if I do that, I will only focus on these classes. So if I flag, for example, leaves as foreground and click on objects, I will segment out the connected components, objects that are from the green class, from the leaves. And you can flag a few of them. So you can also flag the stem, for example, and the virus. And in that way, we are suppressing the background and we are only looking at the foreground. Now, if we do the same thing now in the visualization, we focus the analysis only on the plant itself, not on the background. So what is actually now happening is that we are only dealing with pixels from leaves, stem, and virus. We may also want to put some specific, really different color on the background if we want. So maybe it's better to put it completely black or more transparency or less transparency so that the plant stands out. We may also change the color map, for example, to gray level. That is very useful for spotting the differences by human eye or different type of color range. And here, basically, by playing interactively with the index, we can often spot things that are not really visible directly in the spectral data or any of the bands. So you can see that the equations allow you to define many different common spectral indices like NDVIs, all these different things that, or many of the things that are published in the literature. You can play with this, each of them with the parameters. You can change the spectral ranges. We can use indices that have up to 4 parameters. So that gives us quite some opportunities. So for example, in our case, we would like to highlight the differences between the healthy plant leaves and the virus infected tissue, let's say. And we may try to do that, for example, with this index. You see that here we have leaves that contain some virus infected parts inside. And if we go to visualization, this might be interesting. So far, we were using the auto scaling. That means that if we switch to a different image, and if you observe the value of the minimum and maximum for the range of the index value, they change. They may change depending on what scan we look. And that's often not what we want, because we want to get the same range. So once we are on some image, we can just disable the auto scale, or we can move minimum or maximum. And you can really adjust the range so that some pixels will start getting out of the range. If we move on the image, at the status bar at the bottom, you can see a number, that floating point number. That is actually the index value. And if we go out from the min and max range, we will see these pixels that are labeled into light blue, and they are above. You can switch it, for example, violet. And the same would happen pixels that are actually getting out of range at the bottom. So this allows us to adjust the range so that our plants are, if we use the entire range basically, and then because we do not have auto scaling, we can compare what happens on a different image. If we are happy with the range over a set of images, we can export this information for further processing. So that's what I would like to show you. The first step was building the classifier. Second step was adjusting or playing with the spectral index. And the third one is actually getting the information out for your further analysis. And that's what we can do with this feature extraction functionality. We can extract information from different objects or different types of definition. One would be from objects that are coming from object segmentation, but that's not so convenient here. In many phenotyping projects, we would like to really extract the information from the entire image, or from a germination well, or from something very specific. And that's what you can now do in perClass Mira. We can go to this region definition and basically just draw a region, give it a name, or just not care about giving it a name, but we can define a region in each image that we are interested in. It can be one, it can be multiple. So in the simplest case, we just want the entire scan or the entire plant. And now if we return back to the feature extraction panel, we can say that we are interested in the image regions. And we will represent all foreground pixels. So the foreground pixels are green, orange, or whatever it is, and red. So the leaves, stem, and virus.
The entire plant.
So we are interested in a plant. We are not interested in background and plastic labels. And then we can basically select what to export. So we could export mean spectrum. We could export, in our case, for example, a single scalar value for the index, but we can also export the histogram of the spectral index that we selected. You can see that there are other options, but let's stick with the spectral index. And then we can select a bunch of scans, go to export, and export this functionality to Excel. One thing that actually I forgot to explain, and I should do that, let me go back. We have decisions. we defined our foreground and we want objects, but we are interested actually in dealing with all foreground classes together. So not separately, but together. That's what I have stressed before. So we go to the spectral feature extraction. We go to here and we export the result from these scans. We got the out.xls file, and there actually what we see, we have different scans. We have in each scan, each image, one or more objects. We only defined one. This is actually the just column, row, and width and height of the object. And then here is the information that we extracted, and by default, that's just a histogram of the spectral index. So what we could do is to see see that we have 4 plants, and 2 of them have this specific mode that is corresponding to the virus-infected tissue, and 2 of them don't. So these histograms are comparable, and here we have the bounds, and you can basically do such kind of analysis for maybe a few hundreds of scans, and then do some statistical processing on that. So that's functionality that would be helpful if you are trying to find out differences between, let's say, stressed plants or other kind of issues. So you see that the definition of the spectral index, currently it's from a list of equations. So we are also interested in feedback, what kind of indices people use, and that's clearly doesn't support everything, but it's quite a rich family already, and we are adding more functionality. And what we find very useful about this whole process, also for ourselves, is the possibility to really directly see the feedback. And often we actually use this not only to compute spectral indices, but actually uncover things that were not clear to us in spectral cubes in different contexts. It can be really quite interesting to play with this functionality. So the process was starting from cubes and pixel classifier, then segmenting objects, being able to visualize only on those objects, and then exporting the information for post-processing.